So before we hear from Dr. Eisenberg, I'd like to fill in a little bit more about his illustrious and wonderful career at UCLA. He's devoted his scientific career to understanding the structure and the functions of proteins in both health and in disease. And he's used a wide variety of computational tools. He's used X-ray diffraction, electron microscopy, and so on. And he's been able to determine that many of these diseases are caused by proteins coming together in aggregates that are very abnormal. And in fact, about a dozen years ago, he discovered the atomic structure of the amyloid fiber spine. And the best I can describe it is it comes together in sort of a zipper structure. He'll have really beautiful photos to show you in models and so on. And so this has now been named the steric zipper. And he's been able to determine the atomic structure of this zipper in 90 disease-related steric zippers, which is really a remarkable, remarkable contribution. He's an incredibly productive research scientist, but he's a wonderful and collaborative colleague here at UCLA. He's also been a fantastic mentor, and I had to write this down. He has supervised dozens of undergraduates, over 60 PhD theses, and over 70 postdoctoral fellows, most of whom have gone on to other research institutions where they're carrying on similar and related work to what they did while they were here at UCLA. He has co-authored over 300 peer-reviewed research papers and written two books, one on the structure and properties of water and a text on physical chemistry for the life sciences. His awards and commendations are so numerous that we went ahead and put them onto your announcements so that you can take your time and read them at your leisure. And so now I would like to ask both Dean Washington and Dr. Eisenberg to come up to the podium for a few tokens of our appreciation for him as the recipient of the David Geffen School of Medicine 55th Annual Lectureship. And if you would hand him his plaque.
There's the cameras over there. Want to step out?
Oh, Judy, you need to be in this picture.
Okay. Okay.
And we have a small token of appreciation, which is going to contribute to his laboratory research.
Wait, I saw Lucy on the front row. Do I go to Lucy?
Better hand it to her. Or better than that.
Thank you, David, from all of us.
Thank you so much.
Very welcome.
And to all of you. Thank you, David.
And so without further ado, Dr. David Eisenberg.
Well, to say the least, I'm overwhelmed. I'm hoping I can pull myself together to give a lecture to tell you about some of these things that have been mentioned. First thing, to diffuse all this praise, I have to say, it was really the work of all these people up here. About 20 presently in the lab. The most recent, Hannah and Baker, the last few weeks. Then about 25 former scientists who've now gone on to other institutions who worked on these projects, and then so many UCLA collaborators just on these projects, and collaborators elsewhere. So it's wrong to say this is my work, it's all our work, and I see some of these people here. I'd really like it if you'd all stand up. If your name is on this slide, please stand up. Come on, everybody. I see Duilio Cassio back there. We've worked together for, I think it's 28 years. A lot that goes on in the laboratory just couldn't have happened without Duilio. Michael Savoya, I think 14 years. Daniel Anderson, about 25 years. So what makes a laboratory, of course, are these long-term really dedicated people. So here's my outline. First of all, what is amyloid disease? And then what are each of these groups getting out of it? Well, first of all, what's in it for me? I have to tell you that. What's in it for the pathologists? They really have the biggest payoff. What's in it for patients? What's in it for our society? What's in it for big pharma? What's in it for the amyloid fibers which cause these diseases? And finally, what's in it for amyloid oligomers? I'll get to those later on. So, what is it? These are all diseases associated with protein fibers with several well-defined properties. They bind certain dyes. They have a common morphology, which you see here. They're elongated and unbranched, and they have a similar X-ray pattern. To paraphrase former President Reagan, once you've seen one amyloid fiber, you've seen them all, at least in the electron microscope. Each disease is characterized by a different protein, its own protein, but they all look the same. Here are tau fibers from Alzheimer's disease, alpha-synuclein fibers from Parkinson's disease. I could show you from all the other diseases, and they do look the same. They cluster into plaques in the brain, first noticed by Alzheimer, beginning of the last century. Alzheimer, in 1901, was the assistant director of the Asylum for the Mentally Disturbed and got his title here, Mentally Disturbed and Epileptics in Frankfurt, Germany. During the days, he worked as a psychiatrist in the asylum, helping patients. But at night, he did dissections of brains. He had the theory that there was some organic cause of mental disease, and he looked for it in brains. He had his big breakthrough at the end of 1901 when he admitted a patient, a 51-year-old female patient. We now know her name, August Dieter. She had, in the past year, lost the facility to form new memories. And she'd become fearful of her former friends, including sometimes her husband, who brought her into the asylum. After she was admitted, she declined rapidly in her cognition. She lived for 4 more years, but the final year, she just stayed on her bed, completely silent, grasping her knees to her chest. When she died, Alzheimer autopsied her brain, and in the cerebral cortex, he saw both what he called plaques and tangles that come from these fibers. Within a few months of seeing that, he formed the hypothesis that her psychiatric symptoms came from these plaques and tangles, and he gave a talk before the Society of Southwest German Psychiatrists. At that talk, when he finished, there was no question and no discussion whatsoever, just silence. And I found a translation of the proceedings that day, and the translation says that the chairman broke the silence. He said, "So then, respected colleague Alzheimer, I thank you for your remarks. Clearly there is no desire for discussion." And then went on to the next speaker. So if none of you say anything at the end of my talk, I'm not going to feel so bad about it. Just to give you an idea of the size, it takes about 5,000 of these fibers together to give the thickness of a human hair. So that's why we have to use the electron microscope to see individual fibers. So now what's in it for me? Here you see my father being surprised by his patients on the day he retired. He was a pediatrician in our village where he was admired greatly. And he hoped I'd follow him into medicine, made that very clear to me. But unfortunately, I went to university, and my undergraduate research supervisor, John Edsel, said to me, if you're really interested in helping in human health, well, you should work in biochemistry and molecular biology. I confessed to my medical colleagues, Dr. Geffen and others, I swallowed that hook, line, and sinker. My father did not. And I began to think, he died 18 years ago, and at that time I started to think, what have I contributed to human health? And I just couldn't think of anything. And I was thinking, now if I meet my father in the afterlife, what am I going to say to him? So I looked around for some area that maybe the people in our group could contribute to, and amyloid fibers seemed like a natural one because we studied proteins, and all of these diseases are caused by proteins clumping together to form these fibers. So here are just a list of some of the diseases, each disease characterized by fibers. In the case of Alzheimer, there are 2 different fibers. There's a lot of discussion in the scientific literature of which is the primary one or the more important one. It's not settled yet. In Parkinson's, it's clear there is a definite protein which seems to be, in some sense, the cause of the disease. Diabetes type 2, another amyloid disease, always associated with a protein called amylin or islet amyloid polypeptide. Lou Gehrig's disease, now with about half dozen different proteins in amyloid state. One of the newly recognized diseases, and more amyloid diseases are discovered every year. There are now 40 or 50 of them. A new one you've probably heard of, CTE, or chronic traumatic encephalopathy. That's the disease that professional football players get by ramming their heads into 300-pound opponents. Huntington's disease seems to be amyloid. In the last 3 years, it's been discovered that numerous cancers are amyloid-related disease, where there the protein is p53, the so-called guardian of the genome. And then there are many diseases where there are many fewer numbers of patients, but these are all fatal, terrible diseases. Then there are the prion diseases, like CJD and mad cow disease. These are also amyloid diseases, and these have an extra dimension to them. They're infectious, that one animal or cell can catch them from others. So anyway, it seemed like a great opportunity to maybe do something for human health. That's how we all got into it. Now, for some of these diseases, the so-called systemic diseases, which are included in this group, the fibers seem to be the cause of the disease. They accumulate in tissues and organs and eventually destroy the function of kidneys or heart, liver, and so forth. So for these diseases, and also maybe for cancer, that if you can stop the fibers from forming, maybe that could be a treatment for the disease or prevention for the disease. In other cases, the neurodegenerative diseases, shown up at the top, in this case, it isn't clear that the fibers are so much the cause, because smaller units of the same proteins that contain tens to 50 molecules of these proteins, called oligomers, they seem to be, in many cases, more toxic than the fibers. This is a big debate that is going on among amyloid scientists at the moment. So what's in it for the patients? Well, in Alzheimer's disease, there are over 5 million patients in the United States. That means about one more every 67 seconds. Because we're an aging population, they're expected to be maybe 15 to 20 million by mid-century. Diabetes type 2, there are already 20 million patients, 1.7 million more a year, each year diagnosed. I read on the web that 1/3 of hospital admittees in California have diabetes type 2. I wanted to ask Gene, is that true? Yeah, really, really shocking. Parkinson's disease, there are about 1 million patients, 60,000 more each year. Lou Gehrig's disease, a really horrible disease. All of these are horrible, but this is particularly awful. 27,000 patients in the United States, about 5,600 more every year, and you can see from these numbers that the patients don't live very long on the whole. Then there are a whole bunch of the other amyloidoses, the other diseases, with 1, 2, or 3,000 diagnoses a year in the United States, and every one of these was a fatal, very unpleasant disease. So there's a lot to do. What about for our society? Alzheimer's is already the most expensive disease that we're dealing with in the United States. One estimate is healthcare costs last year were $214 billion, expected to go up to $1 trillion in 2015. $1 trillion is presently 7% of our gross domestic product. So it's clear that this is an unsustainable situation. We have to do something about Alzheimer's disease. Of course, we have a center here headed by Dale Bredesen, where we have a lot of scientists working together on this.
In 2015, right? You don't mean next year it's going to go up fivefold.
No, I meant 2015. I meant 2050. Mid-century, 2050. Thank you, Steve. Yeah. Yeah, things just got a lot better. Another statistic is that there are usually 2 people that are, at least, who are caring, family and friends, are caring for their loved ones, which comes up to about 17 billion hours a year of free care. If you just take the minimum wage, that amounts to another $220 billion that goes into Alzheimer's disease. According to the web, it's so stressful taking care of these patients that 1/3 of caregivers report to medical care with depression. It's the only disease in the top 10 that so far cannot be prevented, slowed, or cured. Diabetes type 2, again, tremendous costs. Again, we have to deal with these diseases. So what's in it for a big pharma? You've noticed they've not been very successful so far at finding drugs for these diseases, despite spending many hundreds of millions of dollars. Why not? Well, one thing is, it's difficult with these diseases of the elderly to find a clinical endpoint. That is, you have a drug, you treat the population, but over the time of the treatment, a lot of those people get other diseases or die. So it's hard to find out whether the drug is working. And that's why in pharma, they're working on biomarkers and diagnoses. Oh, those are very important. Now, from my personal point of view, one failure of pharma has been to ignore structure-based design of drugs. That's worked so well for cancer and metabolic diseases. Why aren't they doing that? So if you ask people in pharma, they say, well, it's impossible to grow crystals of fibers, so we don't try. So how do you treat an impossible problem like this? The answer is you give it to a UCLA graduate student. And this is Melinda Balburney, and she found you can grow crystals of amyloid proteins. What she discovered is that the segments of the proteins, it isn't that the whole protein unwinds and then winds up into a fiber. It's rather that the protein opens, it exposes a short segment, and some of the segments within proteins are very adhesive. They bind to identical segments in other molecules. So here are short segments of one protein, even shorter, and they themselves form the fibers. And then Melinda discovered they also form crystals. But you can see these crystals are very small. This is a light microscope picture, half a millimeter this is. So hold your fingers up half a millimeter. It's very little. And here they're tens or hundreds of thousands of little microscopic crystals. You can see them in the electron microscope, and they have nice faces and edges, and they have a good diffraction pattern, but we have to figure out how to get a structure. So I think Melinda Balbirnie basically made several discoveries. The first is that these adhesive units that aggregate amyloid proteins are short sequence segments. It didn't sink in right away, but over several years we began to realize that. Second, they can be crystallized, giving the chance to get atomic information about the structures. Amyloid crystals turn out to be always microscopic. We don't fully understand why, but it's probably there's a strain that's in the crystal. So here you see fibers growing out of the crystal, and the fibers tend to twist. They're held straight in the crystal, and that limits the size of the crystals, we think. And then I think the most important discovery from Melinda's work is give impossible projects to UCLA researchers. So the next impossible project is determine the structure of these tiny crystals. And there we struggled in the group for several years, 3 or 4 years. But then we learned of Christian Rieckel, who was at the synchrotron in Grenoble, France. And he is a professional there who perfected a microbeam. And you can see the size of this beam is about 1 micrometer, which was comparable to the size of our very largest of the crystals that Melinda Balbirnie was able to grow. And here are 100 of those crystals that were used to determine the first structure in this mosaic. And it's scaled by a dime. You can see ME of dime here. This is a dime. So you see how small the crystals are. And here I've enlarged 4 of them over on the right. You can see most of what's shown here are just glass pins on which the crystal is mounted. So by mounting these tiny crystals on glass pins, you can see the cross-section of the microbeam. Then it was possible to collect data, and then the structure was determined by Rebecca Nelson and by Michael Sawaya, who's here. And this is the structure. And basically, you can see that the protein chains are extended. These are the chains. There are only 7 amino acids long in these adhesive segments. They stack up into sheets. And if you look down the axis of the fiber, like now, you can see the side chains form that zipper that Judith Gasson was describing. We call that a steric zipper. So the fibers have not 5 layers as shown here, but more like 100,000 layers, the fibers that I showed you, and the crystals. And they're made up of pairs of these sheets of proteins held together by hydrogen bonds and then these side chains in the steric zipper. So if we look down the sheet, now we're projecting down through maybe 50 or 100,000 layers. We're looking at a fiber. You can see these side chains, the teeth of the zipper, and how they interdigitate, forming a very tight interface here. The atoms are right up against one another. Outside are water molecules. That's the red pluses. Inside, it's completely dry, forming this tight structure. So that was our first structure published by Rebecca Nelson, Mike Sawaya, Melinda Balbirnie 9 years ago. So since then, as mentioned by Judith Gasson, she said 90. Judith, you're out of date. Now it's about 125 crystal structures of these fiber-like steric zippers in crystals from 15 different disease-related proteins, some from prions, A, beta, and tau, from Alzheimer's, alpha-synuclein from Parkinson's, one of the protein from diabetes type 2, islet amyloid polypeptide, several of the proteins that give ALS, and some of the other amyloidoses, the systemic ones, and p53 cancer. In all cases, we're projecting down the structure through many layers, and they all have 2 sheets, and you can see they're pretty well interdigitated. So now I'm going to give you an example of a very recent structure that was determined, and it's of the toxic amyloid spine of alpha-synuclein, the protein that's associated with Parkinson's disease. It's only 11 residues, but it's a segment of the so-called NAC domain of this protein, which has a total of 140 amino acids, only 11 in this structure. But this segment has been shown by others, Virginia Lee, University of Pennsylvania, and others, to be necessary and sufficient for alpha-synuclein to aggregate into fibers and to be toxic to cells. So it looked like a good bet to learn the structure of. Maybe we can understand from the structure how alpha-synuclein kills cells. So the crystals were grown by Magdalena Ivanova. She's now assistant professor at Michigan. And these crystals were smaller again by a factor of 10,000 from the crystals I showed you before. In fact, they're so small that they're invisible in the optical microscope, because you can see this is 500 nanometers. That's the wavelength of visible light. And so you can't see these crystals in the microscope. So how to cope with that? Well, our wonderful staff stepped up to this, and Jose Rodriguez, a postdoctoral fellow, a Giannini postdoctoral fellow, started going to Janelia Farm, where they have excellent electron microscopes in the lab of Tamir Gonen, and he was able to get electron diffraction data. The structure I'm going to show you is the first unknown protein structure to come from electron diffraction. And from those data, Duilio Cassio and Michael Sawaya were able to determine the structure. And here it is. The structure is this colored part. This is what we see in the crystals. And you can see the steric zipper in the center, and then the chain bends, and it forms another steric zipper-like interaction here. And then the rest of the outside in the white area, well, that's more or less made up. This is a model based on this crystal structure. And now you see another view of it. This is the stacking of this NAC domain. And again, there'd be tens of thousands of layers. But now we know the structure. And what does that do for us? Well, maybe we can learn to inhibit it and test those inhibitors. So that brings me to what we can do with amyloid fibers. What's in it for the fibers? And the goal is to be able to use the structure to design inhibitors. That's the next step in this process. And we start with tau, which is one of the Alzheimer's protein. Again, we identify the short adhesive segment. It has 6 amino acids. This is the one-letter code for my wife and friends here, VQI, VYK. That means to us certain amino acids. And if we just take these 6 amino acids and synthesize this short peptide, well, it forms fibers on its own. It's the adhesive segment. And it also forms these horrible microcrystals, but they're enough to determine the structure. And that was done by Shilpa Sambashivan, graduate student, and Michael Sawaya. And from that structure, it was then possible to design an inhibitor. This inhibitor is a peptide made of D amino acids, and it was designed by Stuart Sievers, who's now a postdoc at Caltech, John Karanikolas, assistant professor at University of Kansas, and in collaboration with David Baker, University of Washington. And this inhibitor, when it's put into a solution of a large segment of tau, now fibers don't grow. And that's shown, first the design principle. It's been automated by Lin Jiang. Lin Jiang's just been offered an assistant professor in residence here in the Department of Neurology. He's a brilliant computational scientist, and he automated this process of designing an inhibitor. And what you see below is one of the sheets of the steric zipper for this tau segment, and on top is this inhibitor, which is being designed, and it's being designed to sit on the top of this steric zipper and prevent other layers from forming. So we can put it into solution with tau, hoping that it'll stop fibers from forming. And that's shown here on this slide. The vertical axis shows the formation of tau fibers over time. This is up 140 hours, so that's about 6 days. The machine had to take a sabbatical on the 7th day. If you shake tau, that's the black curve, it forms fibers, as shown by the fluorescence of a dye that binds to it. But if this inhibitor is put into the solution, then no fibers for up to a week. So it's a lead to a drug. That's the hope, that we'll be able to go from that to a drug to stop the formation of tau fibers. So now I go to one other example, my second last example of designing inhibitors. And this is an inhibitor against fiber formation of the protein p53. p53 is found to be mutated in about half of all cancers. It forms fibers in some of these cancers. We don't yet know the percentage. In many cancers it's not in fiber form, but in many it is. And this amyloid p53, it soaks up all of the p53 in the cell. It's in the fiber form. The guardian of the genome is off the job. Normally, it senses when cells are proliferating, and it sends them into apoptosis. They commit suicide. But when it's off the job, the cells are free to proliferate and make a tumor. So we want to stop its proliferation. Here's a diagram of p53. It's a very complicated protein of 400 amino acid residues. Again, we identify the region which is the short segment which is adhesive, and it is 7 amino acids long, and here's the amino acid sequence. Again, we pronounce this, we call it L-title, and what we want to do is now make an inhibitor to the stacking up of L-title. And here's the idea. When we look at the known structure of p53, determined Memorial Sloan Kettering, that L-title, it's completely buried in p53. But when there's a mutation, this star means a mutation, then this L-title comes out. p53 is a very floppy protein, and many different mutations, this L-title comes out, so it can stack up and form a fiber and take p53 off the job. It's inactive, and that leads to proliferation of cells. So the first thing is to determine the structure, and Alice Saragni's been leading this project. Angie Soriaga determined the structure from material synthesized by Lisa Johnson, and Lin Jiang again designed an inhibitor to go on top and to stop the formation of the fiber. You now recognize the steric zipper, and you know that looking down you see the side chains interacting. So that's old hat to you. So here are the steps that we go through. First, we find out what this adhesive, which segment is the adhesive segment. It's L-title. Then it's synthesized, and we find that it forms fibers. It always does, and almost always forms microcrystals. Again, you see these tiny crystals. This is in a microscope slide. You can see, again, very tiny crystals, but Angie Soriaga was able to determine the structure shown here. And then Lin Jiang designs an inhibitor to go on the top. So that's the next step. And then the inhibitor is tested. It's tested against this L-title. And if the inhibitor is stoichiometric, one inhibitor per one L-title, then you see it doesn't form fibers. This shows fiber formation against time. So the inhibitor can just about stop the formation of the fibers. And then the next step is to test the inhibitor, not just on this segment, but on cells. So to do that, you have to get the inhibitor into cells. So Alice Soriaga coupled the inhibitor to polyarginine, tagged 9 arginine residues with a little linker. It goes into these cancer cells, the cell line. When it does that, it reactivates p53. It sends the cells into apoptosis. You can see they start to die. So it looks good at the level of cells. And then the last step is to test it in animals, in mice. And so that's been done in collaboration with the laboratory of Sanaz Memarzadeh, who's in our stem cell center, and Dr. Diana Jansen, who works with her, again by Alice Saragni. So here's a test in cells looking at how the cells survive after 7 days of treatment with that inhibitor. And several cells that come from patients of Dr. Memarzadeh, the inhibitors tested on those, and p53 seems to be reactivated and the cells die. Two other groups of cells that have p53 that's not in the aggregated state. These are negative controls, and there the inhibitor doesn't seem to do very much. So it looks healthy in cells, like the inhibitor's working. How about in mice? Again, these experiments are done in the laboratory of Sanaz. So a mouse is injected with ovarian cancer cells in the flank, and then it's treated with this inhibitor, 15 milligrams per kilogram, over a period of several weeks. There are 2 controls. The first control is the inhibitor is just scrambled in sequence, so it shouldn't bind to p53, and the other control is just the vehicle, the buffer in which the inhibitor is normally delivered, and it's injected in the peritoneum of the mouse. And here are the results of that experiment. The tumor grows in the mice with the scrambled sequence or with just the vehicle, but with the inhibitor, and now the tumor doesn't grow, it seems to decline. And after 2 weeks, these are the tumors from the mouse that have been treated, and then from the mice that have not been treated. And here are the weights of the tumors. So it seems to reduce tumor growth about 75%. So it looks like maybe, again, this could be a lead for a drug for cancers that are based on aggregated p53. So the inhibitor seems to reactivate p53, both in the cell lines, in patient-derived cells, that carry the aggregated p53. It then somehow allows properly folded p53, which can transcribe downstream targets and then send the cells into apoptosis, drives the cancer cells, and they stop proliferating. In mice, we see inhibited tumor growth. It's specific, that is, with a different sequence. The inhibitor doesn't work, doesn't inhibit tumor growth. So what are the nice things about this inhibitor? First of all, it only is active on cancer cells that contain the aggregated p53. That is, it doesn't do anything to other cells. And Alice Saragni's investigated this. Seems to be no effect on folded active p53, no hyperactivation or increase in normal cells. The other nice thing is, it doesn't depend what the mutation is in p53, and there are many mutations, hundreds of mutations that lead to cancer. One inhibitor will work for all of those, just so p53 goes into the aggregated state in that disease. So it prevents the aggregation, so it should work against all of those mutants. It also blocks aggregation of wild-type p53 with the fibers, and it penetrates into cells and seems reasonably stable apparently owing to its residue composition and sequence. So I might come to my last topic, and that's amyloid oligomers. As I mentioned before, they're somewhat mysterious, but many scientists, many laboratories have found that more toxic to cells than the fibers are smaller units for the neurodegenerative diseases. So finding out about this seemed like an absolutely impossible project. What a wonderful project for a persistent student like Arthur Laganowski. Arthur is now assistant professor at University of Texas. He just started his own lab. You can see him here with his crystallization trays. He set up 140,000 experiments. On top are the 96-well trays, and here are the 24-well trays. And he asked Duilio Cascio to take his photograph with these before they were thrown into the garbage. So what are amyloid oligomers, and how would Arthur find out? Well, it was found by others that they're more toxic than fibers in Alzheimer's disease and some of these other diseases. It was known that there are fewer molecules in oligomers, more like on the order of tens rather than hundreds of thousands, and they're transient. That is, they don't last very long. Oligomers go away and fibers are formed. So how do you study them? That's part of the problem. So Arthur selected a segment from the protein alpha-B-crystalline to find the structure of an oligomer. Alpha-B-crystalline is the main protein in your eye lens, studied here at UCLA by Joe Horowitz for many years. It can form amyloid fibers. It can do in the test tube, and it can in your eye, and then you get a cataract, and you have to remove your lens. So I'm not sure what Arthur was thinking, but maybe it was that it takes decades for it to form an amyloid, so maybe we could trap the oligomer. Maybe he could trap the oligomer. So he was able to grow a crystal, and you'll see this is a real crystal. It isn't one of those miserable tiny needles. So it was evident it was something else. And so it was a segment from alpha-B crystalline of 11 residues, and you can see it forms a barrel, and the barrel has 6 strands, and they're anti-parallel beta strands. There's no structure in all the 100,000 structures in the protein data bank that look like this one. This is the first 6-stranded regular barrel. It is toxic to some extent to cells, not as toxic as A beta. It's under 100th as toxic A beta, but nevertheless, it is toxic, whereas some of our steric zippers are not. If you imagine cutting this and opening it up, so now we have the 6 strands, we've laid them out like this, you can see that unlike the steric zipper where the strands are in register, right on top of one another, here each pair of strands slides along with respect to the last one, so they're out of register when it forms this barrel. We think this might have something to do with the toxicity. You can see that this interface is weaker. It has only 4 hydrogen bonds rather than 6 in the stronger interface. We think that has something to do with toxicity. So this is our present thinking, or hypothesis of amyloid toxicity. If you have a monomer, it can form an in-register beta sheet, like we saw in the steric zippers, and then go on to form 2-sheet steric zippers. So we've seen a lot of these, 125 or so. Occasionally, it could form an out-of-register sheet. Here, the sheet is not as stable. They're dangling hydrogen bonds. So it's a higher energy, which means it's likely to go back to monomer and then go into the fiber state. It could form one of these closed barrel cylindrin, which we know is toxic. So at present, our lab is working on this hypothesis, and we're seeking structures for these toxic amyloid oligomers again, in order to design inhibitors to them. And the furthest along is the protein SOD1, studied by Joan Valentine for many years, which is associated with ALS. And there, Smriti Sangwan and Annie Zhao have discovered that there's a cylindrid-like oligomer found. They've done a structure for that, and it seems to be, it is toxic, and this is a project in collaboration with a Ben Novich lab who make motor neurons, and this is toxic against motor neurons. So we're looking for oligomers in other proteins as well. So what do I hope you remember from this? I'm speaking to my wife, my friends, but also scientists here. There are a lot of patients so far, not with really effective cures. The country's facing a near unbearable healthcare burden. We've got to do something. Structure-based drug design really hasn't been done until now. I think it's a good bet to get going on that. Inhibiting fibril growth can possibly slow or halt some of the amyloid diseases, the systemic diseases, and perhaps these p53 amyloid cancers. Inhibiting amyloid oligomers is probably going to be necessary for some of the neurodegenerative diseases. That's a more difficult problem, but one we're grappling with in the lab. So I'm back again to the great list of people who worked on this. It's been a wonderful experience of mine to work with all of these people, many UCLA collaborators. I am now a consultant to a small company which has licensed some of these discoveries from UCLA and HHMI, trying to take these primitive inhibitors and see if they can start making them into drugs. Although this list of collaborators looks long, I've got to make more of a confession. I went through my papers and looked for all of the collaborators I've had since I came to UCLA, 48 UCLA faculty. So you can see I haven't really done anything. And many of them are here in this room. Some have retired by this time. But it's been a great pleasure working with all of them. And I think this shows, as much as anything, UCLA is a great place to do science. It has a wonderful collaborative spirit. And I thank you for coming today. So are we going to leave in silence, or are there going to be any questions?